So I'm Kristen. You always have to introduce yourself in case you didn't know. And today my talk's all about milk, and I thought this slide was so fitting that milk is the showcase. And hopefully you walk away from this learning a little bit more than you already have. I know many of you have heard my talks before. So my talk title is Unraveling the Complexities of Milk, Production and Composition, an Omics Approach to Infants' First Food. I'm Kristen, and I'm in the Corp Lab, and the NCU is my So why study milk? I remember I was asked this question the first time I presented this project, and many of you know the first time you present any project, it's usually a mess. You don't really know the science as well, you don't know your data all that well, and you haven't seen your own goals yet. And I remember talking to Keith about this, and Keith is saying, well, you know, Kristen, why should people care about this talk? You need to make sure everybody understands your take-home message. Why should they even want to listen in the first place? And he's like, and I'm like, well, you know, the proteins in milk are really cool, and it matters that the genes are expressed and the mammary gut. He's like, no, Kristen, why do people care about milk? And it's because milk saves babies. Babies need milk to survive in most contexts, and really driving into that is that milk throughout mammalia, through humans, all the way down to monotremes, all over the place in mammalian and mammals. And mammal babies need milk, and so milk is about saving babies. And this project, as I was trying to think, okay, how can I give a background of all elements of my work, and it really spans the entire central dogma. In this project, we've looked at gene expression at the onset of lactation. We've looked at transcript-derived protein databases. That's some of the newer stuff, and I'll actually wrap with that last. And then also, of course, the proteomic composition of milk. So it really has hit all elements of that central dogma. So really, why study milk biology? Yes, it saves babies, but are there other reasons? So there are 5 reasons that I want to highlight for you today. One of which is that milk composition shapes infant health. We know that breastfeeding reduces the risk of asthma, diabetes, lowers BMIs of infants later on in their life, and that this nutrition at this early time point is so crucial for the infant's success later on. And in addition to that, milk is a single solution that provides everything that the infant needs. They don't consume anything else as soon as they're born. And it provides all the nutrition in one solution. And to me, this gives us an opportunity to maybe understand adult nutrition better. I know if you've asked 5 people in this room about what perfect nutrition is for adult, you would get 5 different answers. I've shown here the new USDA standards of what your plate should look like, and even this is very much subject to debate and a high moment of controversy. But if you look at some of the compositional elements of milk, maybe we can inform our adult nutrition later on in life and try to figure out what is important for all of us to feel healthy and happy as well. In addition to that, milk gives us information about mammalian evolution. The production of milk has been under very strict Darwinian pressure, or heavy Darwinian pressures, for over 120 million years. That is a long time. And on top of that, milk is produced as an energetic balance between mother and infant needs. The mom is literally decomposing her body, or breaking down her body to feed her infant. So that energetic task in itself, if there are things in milk that are not necessary for the infant, those will be removed through years and years of selective pressures. And rightly so, the things that are necessary for infant will be maintained. And in addition to that, we can also utilize or understand milk, or use milk research to tell us more about different model organisms, such as the rhesus macaque, which is one of the animals we've used in this study. We've also worked with some bovine milk as well, to better give us understandings of differences in primate nutrition, and also to see if through this is something that we can better learn what is uniquely human, or what is something that the humans need more so than other species, to where we can get hints at what might be more beneficial to supplement infant formula. In addition to that, dairy is a $170 billion industry. There's a huge commercial and agricultural relevance to studying milk. As a side note, as I was digging all these images up the other day, it was right before lunch, and I was so hungry, and I was thinking about this. And I was like, I guarantee everybody in this room has had a dairy product in their entire life, whether aside from breastfeeding at some point. We all consume dairy in some capacity, and the omnipresence of milk is huge. And so understanding it from a commercially beneficial standpoint and also an agriculturally relevant standpoint warrants further research in milk. In addition to that, milk is a tool to study for potential disease diagnostics. 2 studies have recently come out showing that there are proteins in milk that are present at differential levels in association with 2 different disease states, one of which is HIV transmission from mother to infant, the other is pregnancy-associated breast cancer. Typically, if you're looking at a new mom and trying to understand anything in her milk, as far as HIV transmission to the infant, or if she does have a tumor, you have to do a mammary biopsy, which is a very invasive thing for any new mother. But if instead of doing these very invasive biopsies, you could instead look at the actual milk composition in some sort of assay, you could better allow the mother to feed the infant in a more beneficial way for the infant, and also just not tax that individual more so than necessary. And truly, there is still so much to learn about milk. And over the last 3 to 4 years, there was really the first time that we've been looking at milk from an omics stance. Some of the papers that I'm looking at are even, like, for example, I'll talk later about some of the RNA in milk. Some of those first transcriptome studies of milk are published less than 5 years ago. And as far as the length of science, that's really new. So using some of these more sequence-based techniques, we can understand milk at a higher resolution than we previously been able to. So one of the first projects that I worked on became termed the dilution effect project. But what I wanted to look at really is what are the effect of some of these high abundance milk protein genes. And we typically were looking at the onset of lactation. So on the left here is a picture of a schematic of a mammary gland and the ductal structures when a mammary gland is not lactating. So you can see the ducts and the branching structures, but as the mammary gland goes to goes into lactation, there's an immense branching and increase in size of these terminal end ducts, terminal ducts on the mammary gland, and the mammary gland undergoes a 70% increase in actual sheer size. And involved in this physiological change are also very significant expressions in gene expression changes. And this is commented in Kimura et al. In 1998, and they start commenting on the dilution effect. This is the earliest reference I could have to this. So the lactating mammary gland produces an enormous amount of milk proteins and their corresponding mRNAs, hence diluting parallel changes in other gene products. It might be for this reason that an upregulation of a specific mRNA or its protein was not evident in the lactating gland. What we found is that as the mammary gland undergoes lactation, there's a huge downregulation of a subset of genes, but also corresponding massive upregulation of things like caseins and whey proteins, or transcripts that end up producing caseins and whey. And modeling this gene expression is a very unique system in that there are no other organs that undergo a similar process, and some of the typical differential expression mechanisms are not necessarily sufficient to model the changes that occur in lactation. And the way I can present this to you as an analogy is picture yourself at your favorite baseball, basketball, hockey, whatever game, and you are cheering with all your might, and you are just so happy that your team's about to score, and you're screaming and yelling. But then you really realize everybody in the room, everybody in that stand, and there might be this super fan that is so loud, and that you may be speaking at your highest voice, but this super fan is definitely making himself heard. And that's kind of what's going on as far as these genes. For example, the individual on the back, where he is yelling at his highest voice, his voice is upregulated as possible, right? But the person in the front is overshadowing that by the fact that they are just so much louder. And that's what we've done to model this dilution effect. And to show you this in actual data form, so this is what I've done here is I've plotted every single gene that's present, their gene expression values for cow and human data. Each dot represents the value that counts of the transcripts present in a sample, and they're just placed randomly. In the next slide, they are placed in the exact same location. And what you see here is there are a subset of genes that go through a massive expansion of gene expression. So the circle here is representative of their expression value. And these highly abundant genes are what's causing the traditional methods of differential expression to be convoluted. So what we found is that there are going to be a subset of genes that are, of course, downregulated between a non-lactating and a lactating state, or non-differentially expressed, or upregulated between the two. But we all, and this is for human. But in addition, let's see here. So, and if we compare that, yes, sorry, I'm mixing my graphs up. So if we look at when we applied our adjustment factor that accounts for the dilution effect, we see that there are a subset of genes here shown in this light orange color that change their regulation status. So they go from being categorized as non-differentially expressed, having an adjusted log 2 value of less than 1, to being upregulated. This was present in human as well as in our cow data. And this subset here, as I said, this is actually what is changing their status to become upregulated. And so what we did is we said, okay, so if these are changing their regulation status, how can we determine if that's accurate or not? And so we compared in the unadjusted, which were the non-differentially expressed, and this is just for example, to the adjusted, and we can compare, do these genes that are reclassified as upregulated, do they look like other upregulated genes, right? Or do they look like other unadjusted genes? And try to see, aside from just their regulation status, are there other characteristics that demonstrate these are truly being misclassified? And what we did is we looked at different enriched GO terms and transcription factor binding sites, and both of these supported the reclassification of these genes as upregulated. And in addition to that, we also looked at histone modifications for gene activation and gene expression, and these also supported that non-differentially expressed genes are better classified. The subset of those non-differentially expressed genes are better classified as upregulated after the application of this dilution adjustment. So what the take-home messages from this portion is that a handful of milk protein genes are highly expressed. Their expression alters interpretation of differential expression, and adjusting those low abundance genes allows for an improved classification of their regulation status and this unique physiological change of lactogenesis. So the next step, or the next kind of chapter of my graduate career was looking at milk proteomics, and many of you probably have heard me talk the most about milk proteins. And so if you break down milk by percentage, it's 31% lactose and human milk oligosaccharides, 31% lipids, and 6% proteins. And each one of these elements have a functional role for the infant, whether it's to serve as an energetic source, whether these are foods for the gut microbiome, lipids are involved in neurodevelopment, etc. And these proteins are really what we chose to focus on closer, partly because there are a lot of issues that come with milk proteomics due to the fact that it has all these other components in it. And so if we were to look at all of the total proteins in the UniPro database, you can see that there's 70,000 proteins in the human reference proteome. Not my studies, but in our lab's previous studies, we found that there are about 20,000 genes that are being expressed. And other studies have shown that mRNA and protein will correlate about 0.4 to 0.7, or R-squared is 0.4 to 0.7. And so with that estimation, about 10,000 of these should have some protein product in milk. So let's see how we've done with them. So about 25 years ago, Kuntz and Lauderdale published the first milk proteome with 7 proteins total. And in 2014, Zhang et al. Published another milk, up till the most recent point, the most advanced milk proteome of 1,300 proteins. So this still falls drastically short of what we would expect to be as far as the total number of protein products. And for the macaque milk proteome, there were only 6 proteins in the total proteome. So there is definitely room for growth and room to better identify and annotate milk proteins. So what we had to do is develop better methods to identify a comprehensive milk proteome. And so briefly, since I know many of you have seen this slide over and over, is we looked at whole milk, and instead of doing a lot of different fractionation steps in the sample prep by extracting the lipids or extracting the sugars, prior to running them on the gel, we actually just worked with the whole gel and allowed the SDS page to clean up those other elements that can interfere with the mass spectrometry at the end of this protocol. We also did gel slicing, which isolates the proteins by their size, and will also keep some of those highly abundant proteins in a single gel slice and a single mass spec run, so that they don't overwhelm the detector and inhibit the detection of some of the more minor abundance proteins. And we also incorporated a glycan cleavage with PNGSF, and this is the first use of this in milk proteomics, and I think this is one of the reasons this protocol has been so successful, is that milk proteins are very heavily glycosylated. However, those glycosylations can interfere with the way they're detected on the mass spec, and by removing those, you expose more of the peptide chain and improve the ability to detect milk proteins. So how well did it work? So for macaque, as you remember, there were 6 proteins previously identified in the macaque milk study. We were able to identify 518. So a humongous expansion of what was previously known for macaque, and one of the first macaque milk proteomes. This is something that can still very much be better improved, as the macaque reference proteome through UniPro or Ensemble is still very poorly developed. So this is still room for improvement in the future. So very exciting results of that. So for the previous human milk proteome, as I mentioned, there was 1,333 proteins that have been identified, and we were able to identify 1,606 proteins. So again, able to identify more milk proteins for human milk. And I worked with one of our undergrads to merge 10 different papers of the most recent other proteomes. Some of these were targeting casein fraction, some of these were targeting the milk fat globule membrane, and we combined all of these and we said, okay, what are all the other proteins? Are we finding anything new? And so all total, for these other 10 papers, there were 2,098 milk proteins, with each having a unique UniPro identifier. And we compared that to our list, and we were able to find that we were able to identify 524 new milk proteins. So not only were we able to detect many of the other proteins previously found, we were also able to build on the total protein catalog of milk. So does our method improve detection of proteins with certain characteristics? We pulled one of the studies, publication Gao et al. That came out in 2012, and tried to compare our protein list to one another. The reason we chose this paper is they have a lot of similarities in our method. They both do a gel electrophoresis. They do gel slicing as well. A lot of things were similar between our methods. However, things such as the PNGSF treatment was unique to our method, and also the single running the whole milk on the gel opposed to subfractionating early on, that was also unique to our method. So this is a distribution of the proteins that were identified by them only, proteins that were identified by both, and it's their protein size by distribution. And if we add in the proteins that we were able to uniquely identify that they missed, we find that there are a lot more proteins with a very low molecular weight. And so we were able to better detect small proteins. And on top of that, this is even more beneficial as far as milk goes, because a lot of the immune proteins are very small in molecular weight. So by being able to better detect small proteins, we can better annotate some of the role that milk has in infant's immune development. And in addition to that, we also found that we were able to detect the proteins that we found with comparable quality. This is the percent coverage of the entire amino acid, and you can see these distributions are pretty similar, and that we were able to get the same kind of quality of our proteome as theirs. And this is the number of unique peptides per protein, and again, comparable distributions. And actually, as I was writing this up as part of my dissertation, I was just thinking, I was like, okay, so what was their total mass spec cost like compared to ours? And theirs was like $15,000 or something, and ours was like $5,000. Like, it was totally different. Like, for example, so we were way less expensive, which is great, and with higher efficiency, and that's a very beneficial thing to any researcher. So the take-home messages from the milk proteome part is that our novel biochemical method identifies more milk proteins for human and macaque. We also have increased identification of low molecular weight proteins, and our more efficient methods requires fewer mass spec runs. So So this is something, and then milk composition differences between human and macaque. So now that we have these two proteomes, how can we compare what is present in either species milk? And typically milk proteomics, cross-species comparisons of milk exist at the macronutrient concentration percentage. So what percent of lipids are there, what percent of proteins are there, but that really neglects the diversity of molecules that are present in this milk and some of the unique functional elements that these proteins can have. So we wanted to look a little closer at how are these species milk different. And so what we did is we clustered orthologs. So if any of you guys have seen me present, I usually say, okay, we found this one unique protein, but there's kind of a discord between the human and macaque milk proteome databases. And so instead of trying to find what is unique between one of the two, we said, okay, if we can find the same proteins in both species, can we see if these are present at different levels? If one might be more important to one species milk compared to another. And so in order to do that, we determined what the protein orthologs are. We used a reciprocal best match BLAST P approach and identified 396 orthologs with a very high sequence identity. So proteins that should be more or less the same between the two. And then we plotted what the differential abundance between the two are. So if they're blue here, this has a higher abundance in macaque, and in green has a higher abundance in human. And some of the macaque proteins that were differentially abundant are things like cathepsin Z, which is a proteinase, and also beta casein, which is involved in calcium ion binding. And the macronutrient concentration of both protein and calcium are higher in macaque. So this fits what we already had understood from the literature as far as some of those macronutrient concentrations. And then in addition to that, we probably noticed, and what stood out to most of you first, is that the majority of all of the differentially abundant proteins were more abundant in human milk. And so we looked at what these were doing functionally, and we actually pulled in Bill Lauderdale, who's been publishing milk protein papers since I was born, literally, and Katie Hine, who's an expert in macaque milk, to see what some of these proteins are doing. So I want to highlight just a handful of these and what we know about some of their functions. So there were a subset of proteins that were enriched in human milk compared to macaque milk due to their role as digestive assistants. They were bile salt simulated lipase and alpha-1 antitrypsin. And then also proteins that were involved in macronutrient processing and immune function. So a lot of proteins that are serving to give infants an extra boost when you're talking about their development and helping them pattern their physiology for further health success. We also looked at proteins and their tissue enrichment to see if there are any regions of the body that these proteins are more enriched for. And this is the list of statistically significant tissue enrichment. As you notice, I've highlighted here in red, these structures are all in the cranial region, the globus pallidus, the adrenal cortex, so on and so forth. So it may indicate that some of the proteins that are more abundant in human milk compared to macaque milk are serving a role in neurological development. So there are 96 proteins, orthologs present in human and macaque milk, and the most differentially abundant proteins are increased in human milk. And also those proteins that are increased in human milk are providing, or may putatively be providing, a developmental boost for the infant. And so one of the last slides that I've been working on very rapidly over this last few months is building out some RNA-seq guided proteomics methods and using those to better improve protein identification. So one of the biggest limitations of proteomics in general right now is that in order to identify a protein, you must have a protein sequence in the reference database. And things like shotgun proteomics that are blind to any targeting almost are missing out in a way because you have to have an idea of what your sequence is beforehand, even if it's not necessarily a targeted approach. So the data analysis pipeline, just for background for you guys, is you get your spectra from, for example, an LCMS or a tandem spectrum like we did, and then you'll also incorporate a protein database. And from that, you push this into a program called Xtandem. This is one of a couple different ones that you could use. But what Xtandem does is it matches these spectra in their ion fingerprint. It matches these spectra to known peptides and peptides from that protein database. These are then subsequently filtered through scaffold that'll match and filter on protein and peptide false discovery rates. So it serves as filtering step. And then from that, you get your protein sequence and your proteome. And the thing that we came to realize, and that Danielle really had the idea of a while ago, is that milk actually contains RNA. So this is a picture here of a mammary epithelial cell, and in gray, you can see lipid droplets. And as these lipid droplets aggregate and pass through the cytoplasm, they bud off, and some of these are captured in have a cytoplasmic crescent. And in this cytoplasmic crescent, it's been shown that there's RNA. And for those of you who are more image focused, this is again, these are the lipid droplets. This is an orange stain of whole milk, human whole milk. These gray blobs are the lipid droplets, yellows are the nucleated cells, and then you can see here in this reddish-orange color is the RNA that's present in the cytoplasmic crescents. And Danielle in our lab, she showed that this actually is a better proxy for milk, or this is an excellent proxy for mammary glands compared to any other isolates, if you were just to look at milk fat, for example, or anything like that. So we now can use the RNA that's in milk in a very non-invasive way to, again, better understand the mammary epithelial cells and also better understand milk composition. And so the last leg of what I have been doing is to really utilize this RNA sequencing data to improve our protein imitation. And so for our work, we have biologically mapped samples where we have a proteomic and transcriptomic sample from the exact same individual, which is very advantageous for this work. And so we have 3 human samples and 5 macaque samples at paired lactation stages, both at peak lactation. And so what I've been building out is a framework to flexibly analyze the best, or to better supplement your proteomics data set. So in this, it's a little bit wordy on this. This was a figure from my dissertation, but I haven't had a chance to make it a good presentation yet, so I apologize that it's so wordy. But the 4 main methods that we're using is a variant annotated FASTA file, RPK unfiltered FASTA file, and a genome guided assembly and a de novo assembly. And so basically what you can take here is you can take your mRNA reads and align reads to a reference using a program like TopHat, and then you can call a variant file where you say, okay, which of these sequences, which one of these genomic sequences are different from a reference, and use that to then generate your single amino acid polymorphisms. We also have been using a program called Custom ProDB, which will take in the reads present in that transcriptome sample. So you can take the entire human UniPro reference and then say, okay, of these, how many do we have transcript evidence for? And filter that down based on the fact that these are expressed in the same sample. So through that, we have the variant annotated and RPKM filtered, so it's gene expression filtered, and then also the just RPKM filtered in itself. The other 2 methods that we're currently in the process of testing is a genome-guided assembly, transcriptome assembly, and then also a de novo assembly, transcriptome assembly. And then both of those undergo a 6-frame open reading translation and are then traditionally processed by adding decoy sequences where the N and C terminus are flipped to help with calculating the false discovery rate, and then also assigning spectra, and then you can assign the spectra and peptides to proteins. And so this is very much something that's still in the testing phases, but some of the work that we really are excited about right now. So in total, so this is the percent spectra identified, and on the left-hand side here you have the traditional run, the run that we ran initially, and this is the control, the reference human and reference macaque. And as you can see here, the percent spectra identified. This means of all the spectra detected, how many of those are usable. And the percent spectra identified for just a UniPro reference database is fairly low, 20%, and I'm sure that's like 8%. But when we look at the transcriptome-derived databases, this is a much higher number of percent of spectra that we were able to use. We also looked at this is the traditional one-step method where you have all of your spectra, you match it against a database, a protein database, and you see what comes out. About a year ago or 2 years ago, I want to say, there was a publication in Nature by Kim et al. and they looked at a proteogenomic approach, not a proteotranscriptomic, but a proteogenomic approach, and they used a two-step method. And so we also tested all of our unmatched spectra. So we take from this reference, those original runs, if we take the spectra that didn't match against the traditional reference and say, okay, how many of those can we match in the secondary step? And in that two-step method, we are still able to achieve fairly high number of total spectra that are identified. And we also looked at the total number of proteins that are identified. So again, you have for human, the reference, which is our control, and then the all spectra, which are either RPKM or variant column. And they reach comparable totals. They don't exceed what we previously detected, and we actually thought they would. But this is something that we're going to have to look into a little closer as to why they aren't matching any higher number. But it could be that you have more confidence in the proteome, even though the numbers are a little lower than what you initially expected. But if you look at what we were able to identify from the unmatched spectra against our proteome databases, there's still a couple hundred more proteins that are able to be identified in the transcriptome-derived methods. And the other interesting thing is when we looked at these variant protein sequences, some of these are single amino acid changes, all of these are non-synonymous, and some of them are multiple amino acid changes. And interestingly, in proteins like lactoferrin and alpha S1 casein, some of the most prevalent proteins in milk, the variant that we were able to detect actually had more spectra assigned to that protein compared to the reference sequence. So by incorporating those variants into our analysis, we're able to better identify differences in milk than the reference. And so that's pretty exciting to see. And then again, in macaque, you can see that the total number of proteins does not exceed what our reference database for if you match all spectra against the protein databases. But the unmatched spectra are able to identify a decent number of more proteins as well. So furthering and increasing the annotation of the proteins. And some of these proteins will be proteins that have previously been detected, but now you can use that spectra. But I would also expect that some of these proteins may not have been previously caught before. So some of the take-home messages from this section. So RNA-seq-guided proteomics provides an alternative to reference databases for non-model organisms. And some of these may be able to outperform if your proteome reference is really not strong. And then also protein variants are present in milk and worthy of exploration. And also testing unmatched spectra in a second pass may improve protein identification as a whole. So in summary, for this crazy work of life, so gene expression changes are affected by abundant milk proteins during lactogenesis, and those have been modeled. Milk protein contents for human and macaque have been greatly expanded, and a framework for leveraging RNA-seq data to overcome one of the most persistent barriers to proteomics has been initiated as well. And so what my next step is to I took a position at IBM where I'll be doing research on the metagenomics and metatranscriptomics of the food supply chain in an attempt to better and earlier and with a lot more data detect foodborne illness. So, we actually move tomorrow, which is pretty crazy. And so for acknowledgements, I would like to thank the dream team in the core lab. So I have had the privilege of being mentored by I've had many mentors, but in our lab being mentored by 3 people who are very different in each of their own ways, bring a very unique skill set in each of their own ways, and have played an amazing role in my life. I thank Ian for introducing me to computation in total and for piquing my interest in genomics and bioinformatics and everything. I think like a different person, and I guess that's what grad school is supposed to do, so thank you for that. Danielle, who couldn't be here today, she is at a conference in Montana that is live tweeting under the hashtag #MilkInMontana, if you want to follow it. But Danielle has been my day-to-day email, I have no clue what I'm doing, please tell me more person, and she has been so patient and so kind and has really pushed me to be a stronger person. Sometimes I remember I was looking at orthologs and I was like, okay, Danielle, how do I find the orthologs? And she wrote back to me and she goes, Google it. And I'm like, what? That was so harsh. And I was like so devastated. And then I Googled it and there's like a consortium to decide what orthologs are. Like, it was this very complicated question and I was just asking her for the answer. But like that kind of response made me be like, oh, I can look things up. I can ask myself. I can figure this out, you know? And Keith, who I've shared my cube with for the last, or who he shared his cube because I came inside. Sorry, Keith, no one want me to be precise. Who I've gotten to share a workspace with for the last 4 years has been so instrumental in teaching me everything so much in presentation and coding and so much. But
not remaining stuck.
Yeah.
Keep it to 5 sentences.
Our lab mates, of course, are amazing. Very hard workers. Chantel and Debbie have worked on this project. They're not pictured in that picture, but very awesome people. My dissertation committee and faculty advisor, Colleen and Angela, for their feedback through my qualifying exam and also on my dissertation. See, the thing is, you just cry and then you have to wrap it up really quick and then get out of time. All of our collaborators, we're working with people in Cincinnati, in Baylor, on campus, at Harvard, all over the place. Of course, our funding sources, you can't have research without being paid for. And the funding source is shown in the center there is the biotechnology program. And they have been much more than a funding source. So I appreciate your mentorship over the years. And my family, which no student has made it through grad school without the support of their family. And of course, my wife, the force behind the curtain. She has been so much strength in those stressful times, and even she understood my research and all that, but always willing to support me and make me laugh. And if you know her at all, you know that I had to search for a long time to get a picture of her not making a silly face. So she has been my rock through much of this. And thank you all for your attention. You guys can ask questions even though I have tears I can handle.
Chris, come. Okay, I'll ask a question. When you were looking at the differences in macaque and human milk protein abundance, how do you adjust for like relative lactation stage? You know, to make sure you're kind of in the same window of lactation.
So we controlled for that. So the peak lactation for humans is at day 180, and in macaques it's day 90, and that's just based off of their total lactation stage in general. And then when we were looking at the spectral differences between the two, we normalized by run. We did a trim mean normalization by run, but we actually didn't adjust for anything like protein content or protein concentration between the two individuals. Partly because we didn't want to artificially bias our quantitation, and also partly because macaques have actually a higher concentration of protein in total. But as you saw, most of the proteins were more abundant in human anyways. So if anything, the proteins that were presented as differentially abundant are more conservative than one would expect if you had adjusted for that protein concentration.
And then one more question, those little droplets, the ones that have this, like the RNA.
The crescent, yeah.
Yeah, so are there non-coding RNAs, and do you think they can influence gene expression?
Yeah, so I don't know for sure about non-coding RNAs. I know there are other like microRNA structures, and that's something that Danielle is starting to look into and seeing, can we model these? Are they having any tertiary structure? And that's a very hard thing to figure out, but that is something that we're able to look into, and now just at this stage we're really beginning to have that ability. And the paper with the RNA-seq or the RNA-seq from milk alone was just a couple years ago. So this is something that I know is definitely on Danielle's to-do list, and I'm sure many other researchers as well.
I feel like I saw you talk about this in the previous talk, but so you had an N of 3 with the ladies. How do you think that would change if you expanded that massively and
Yeah.
different ethnic backgrounds and stages of life and diet and all that kind of thing?
Yeah, so you're likely viewer number 2. But I would definitely, there is variation between individuals. We saw that even within our 3 samples. The pregnancy-associated breast milk sample study, they had, I can't remember what their N was, maybe like 10 or 12 moms, but we looked at some of their data and they had variation between individuals as well. For ours, they were all on the same diet, omnivorous, and weren't taking any medications. We tried to stratify by ethnicity, partly because we wanted to get as many proteins, even if they're different, as possible, because our goal wasn't necessarily like, this is the milk protein, like the consensus milk protein. We were saying like, these are all proteins that have ever been in milk. And so we had a Hispanic American mother, an African American mother, and a Caucasian mother. So different ethnicities, and there were differences between them all.
Was there a particular class of protein that was highly conserved versus not? I mean, I know it's a small end. But...
Yeah, we didn't really look at that particularly. There was one mom who just had way more protein in general, and actually, I don't know which number mom she was. I think it might be the Caucasian mom, but I can't completely remember. But her identifier was HSA 1063, and she had a lot more protein in her mom.
Question. So after your second step process to identify more spectra, how many spectra remain unmatched as like a proportion? And is there anything that can be done with it? Will there always be some of those spectra which we can never do anything with?
So the best two-step method, or the best step, well, you're saying on a two-step specifically, right? That's what you're asking, or on either.
Yeah. Just in general, how many spectra do we play or to match? Do you think we can make some inroads?
So on the percent spectrum that match in the two-step, it matches about 50% of those, but you're still talking about 10,000 or so spectra. So there's about 5,000 that aren't matching in the two-step method. So after everything's matched in the one step, and then you have the remaining, there's about 5,000 aren't. Some of those will not match due to quality, and some of those will not match due to like partial cleavages. So prior to going into the mass spec, these samples all these proteins are cleaved to lysines and arginines with trypsin. And sometimes if there's a partial cleavage, it'll mess up basically the ion fragmentation. And so it could just be a poor spectra. The other thing is it might just not have enough intensity to be valid, or to be of a good enough quality to be assigned to a peptide.
So some amino acids like leucine can stimulate growth a lot more than other amino acids, acting as kind of signaling molecules. Have you examined the amino acid composition of milk to see if it is different from general human protein?
Yeah, so we haven't looked at that between species or even between individuals. When we were looking at like I mentioned, that one mom had way more protein in her milk. We did do a AAA test to look at the amino acid composition, but we didn't it breaks down by amino acid level, but we didn't look at, for example, leucine specifically. But there is variation in protein concentration between other, but we didn't break it down to the point of amino acid. But I think that would be something interesting to look at.
What are some potential applications, let's say, way down the road? I know this is very early stage, but could you envision, I don't know, just some thoughts?
I think one of the big things that would be very interesting would be applying this proteomics method, or even some of the RNA-seq stuff, to those two disease states that I mentioned, where we already have evidence that there are proteins at differential levels for pregnancy-associated breast cancer or HIV transmission from mother to infant. And especially with the HIV transmission from mother to infant, in developing nations, if you could detect that earlier on and let a mother feed a child who otherwise wouldn't have a stable nutrition source, that's very beneficial. So I think an applied method from that would be good. And then also just the fact that you can do this small volume is great for other primates in captivity. If you need to look at what's in their milk, I don't know if they're, and then also you can always look at like neonatal stuff, or premature babies, just having a method where it's a very small volume that's required, but with high throughput, that's advantageous for a lot of different types of samples. I'm 2 minutes shy of 1 hour. Thank you.